All right, let's talk about one of those questions. You know the one, a big, beefy 11-mark AQA biology question on DNA that just seems designed to trip you up. Well, today, we're going to crack it. We're going to go through it piece by piece, and I'm going to show you the exact words, the exact calculations that the examiners are looking for to give you full credit. Ready? Let's dive in.
So, what's our plan of attack? First, we'll break down the question itself. Then, we'll absolutely nail the definitions and descriptions. After that, we tackle the main event, that monster 5-mark calculation. We'll grab an easy final mark on non-coding DNA, and then I'll give you a full marks answer guide you can use for revision. Let's get to it.
Okay, first things first, we've got to deconstruct this question. Before you even pick up your pen, you have to know what this 11-mark mission is actually asking for. It's all about understanding the game before you play. And here it is, in all its glory. This is from a higher tier paper, and you can see it split into 4 parts. We've got a definition, a description, a pretty hefty calculation, and then a final bit on gene expression. There are 11 marks just sitting there waiting for us. Our mission is to walk away with every single one.
Right, let's get some easy points on the board. These first couple of questions are all about getting those basic definitions perfect. Don't underestimate them. Nailing these builds momentum and confidence for the tougher parts coming up. So, question 4.1 is just 1 mark, but look at that command word, why. That's a huge clue. The examiner doesn't just want to know what a polymer is, they want to know why DNA is a perfect example of one. And here's the money answer straight out of the mark scheme. DNA is a polymer because it's a long chain made of many repeating units. Simple as that. You could also say many joined nucleotides, because nucleotides are the repeating units, the monomers of DNA. The key idea here is repeating units in a long chain. That's your mark.
Now we're stepping it up. 4 whole marks to describe a nucleotide structure. That word describe means we need to say what it's like. We need to paint a picture with our words and list its components.
For your first mark, you just need to state that every nucleotide has a phosphate group. That's it. Write that down and you're already on the scoreboard. For mark number 2, you say that this phosphate is attached to a sugar. If you want to show off a little, you can specify that it's a deoxyribose sugar. That's a great detail to include. And the third mark comes from saying that the sugar is then attached to a base. So you've got this little chain, phosphate, sugar, base. That's the core of it. 3 marks down. So how do we get that 4th and final mark? Well, you just have to say that there are 4 different bases it could be, and to be absolutely sure you get the mark, name them: adenine, cytosine, guanine, and thymine, A, C, G, and T. And just like that, that's 4 marks in the bag. Okay, deep breath. This is the big one, the 5-mark calculation. This is where you can really pull ahead of the pack, and the secret weapon here is showing you're working. Every single step. Let's go back to the question for a second. See this diagram? It gives us the most important clue for this entire calculation. It says the length increases by 0.34 nanometers for every pair of nucleotides. Circle that word, underline it, whatever you need to do. That word, pair, is everything. So let's break it down into 3 simple steps. Step 1, we need to find the number of pairs. The question gives us the total number of single nucleotides, 1.2 times 10 to the 10. So to get pairs, we just slice that number in half, which gives us 6 times 10 to the 9 pairs. Step 2, we calculate the total length in nanometers. We take our number of pairs and multiply it by that magic number, 0.34. That works out to 2.04 times 10 to the 9 nanometers. And finally, step 3, we convert to meters. There are 1 billion nanometers in a meter, so we divide by 1 times 10 to the 9. And that leaves us with our final answer. But hang on a second. Let's pause here, because this is the number 1 spot where people lose marks. Why on earth did we divide by 2 at the very beginning? This is a classic exam trap, and we're not going to fall for it. Here's the deal. That 0.34 nanometers value is for a pair of nucleotides. You know, 1 on each side of the double helix. But the question gave us the total number of single nucleotides. So before we can use that 0.34 value, we have to figure out how many pairs we're working with. It's a tiny step, but it's worth a mark, and it's the key to getting the whole thing right. And there you have it, 2.04 meters. Isn't that wild? If you could unravel the DNA from just one of your cells, it would stretch out to be over 2 meters long. That's taller than most people, and it's the correct answer for a whopping 5 marks.
We are on the home stretch. We've defined our terms, we've crushed that big calculation. Now, let's just scoop up this last beautiful, easy mark.
Okay, the last part of the question. It asks how non-coding DNA, so the bits that aren't actual recipes for proteins, can affect how genes are expressed. Gene expression is basically the process of turning a gene on. So what's the role of this other DNA?
And the answer couldn't be simpler. You ready? For 1 full mark, all you need to say is, non-coding parts of DNA can switch genes on or off. That's it. They're like the light switches for our genes. An easy mark to finish strong.
And that's that, 11 out of 11 marks. To make sure all of this really sticks in your brain for exam day, let's wrap it all up in a neat little summary guide.
Think of this as your blueprint for a perfect score. For the polymer question, use the phrase long chain made of repeating units. For the nucleotide, remember the sequence. Phosphate attached to a sugar, which is attached to a base. For the calculation, it's a simple process. Half the total nucleotides, multiply by 0.34, then convert to meters. And for non-coding DNA, the killer phrase is switch genes on or off. These are literally the phrases the examiners are looking to tick off on their mark scheme. So there you have it. That is how you take a big, intimidating exam question and break it down into manageable chunks to get every single mark. You've now got the blueprint for this DNA problem. So the real question is, what other tough science questions do you want to see us crack open next?